Okay, I think we're gonna get started. We're 1 minute past. So today, Dr. Jenna Rich is gonna present to us. Dr. Jenna Rich is a postdoc with the Mizzou Animal Genomics Group, where she will help create the first non-human genome in a bottle resources started with cattle. Her work provides computational solutions for studying comparative, population, and conservation genomics. She did some undergraduate research in wildlife disease ecology, and that gave her a passion for One Health, which is the interconnectedness between plants, animals, and people. Outside the lab, she enjoys spending time with her two cats, dog, and her husband. And with that, Dr. Rich, the floor is yours.
Thank you, Josue. And again, let me go ahead and start sharing, and then we can get started. All right, can you see everything okay? Yes. Thank you again to Josue and Meng for inviting me to speak today. It's an honor, and I'm very excited to talk more about my work with deep variant. So my plan for today is a little bit different. So I gave a talk that Josue and Meng attended at LGBT Ag, which I will briefly go through as a review of everything we've learned with cattle. And then my hope is that we can sort of use my method slides at the end as more of a discussion for any questions you may have about how to potentially do this type of work in your species. And then depending on time, we have a little bit of a demonstration. So my goal for today is to primarily convince you of one main point, that deep variant is superior to GATK. However, there are consequences when we use a deep learning model that has been exclusively trained with human genomes. And the most common question I get when I talk about this work is, what about blank or my favorite species? So hopefully I will address that question for you today. And then lastly, I want to leave you with this new idea that we've had or we're working towards, which for any Lord of the Rings fans will recognize this reference. We're calling it one model to rule them all, which is basically just this golden generalizable model. And to get to this conclusion and to get to these ideas, we had to go on a somewhat long and arduous journey. So I want to first introduce the cast of characters, as I like to call them. So in my world, everything is animal focused. And so although many of our colleagues and even internally, we work with lots of different species, but the common challenge that we face is usually due to the fact that we're not working with human genomes. For me personally, I'm also interested in this gradient between domesticated, wild, and endangered species. But there are some pretty obvious challenges that arise as soon as you want to work in those species, especially when you're trying to use GATK. And this is because VQSR, one of the key important steps during that process, does not work very well with small data sets, which are very common with these wild or endangered species. Lastly, GATK and the documentation always emphasizes the fact that the best practices are intended specifically for human genomes. And while they're commonly adapted and customized to work with another organism, there's also varying degrees of effort to make that happen. And then very commonly, lots of different deviations or different practices, and not a whole lot of standardization, and therefore different levels of success optimizing these tools for optimizing GATK. And we see this when we look at our data in cattle. So we know that the optimal or the defaults are optimal in humans, and what happens when we change them. So different VQSR parameters can drastically shift the number of high quality variants that we get. So here you can see in bovine and bison genomes that just changing the parameters during VQSR can lead to a difference of 60 million different variants at the exact same quality. So the underlying data aren't changing, it's just the VQSR parameters. And so obviously when we first heard about deep variant, we were very excited. So in continuing my Lord of the Rings theme, I refer to GATK as sort of the Shire. It's the safe and familiar, comfortable place. And deep variant is this new and shiny Rivendell that is, technologically advanced potentially. So when deep variant came out and they demonstrated how more superior it was in humans, this was obviously very exciting to us. And our initial assumption when we began this project several years ago now was that the deep variant model would just work natively in any of the species we gave it. And as we began this work, we quickly realized that that assumption was wrong. So assuming that's correct, then the next question is, are we going to end up having to retrain this for every single one of the species that we work in? And I'd like to propose that, or we are starting to propose this idea that perhaps we could create this universal golden one model to rule them all. So again, today all of everything I'm going to be talking about is in cattle and the outgroup species, but I want to just reiterate that we're using cattle as a roadmap for all of the other species that we work with. And for me personally, I want to be able to use these tools in endangered species. So before we get into the meat of my presentation, I just want to refresh what makes a good deep variant model. So the common metric that is typically used is known as F1 score, which is simply the harmonic mean between precision and recall. And a 1 value is typically treated as a model that has made a perfect prediction of the truth labels. And in humans, this is very easy to do, well, relatively speaking, easy to do because they already have the genome and model resources, which are just a community standard that everybody decides means is the truth. In cattle and any other species, we don't have those yet. So we're kind of inferring truth based on our high quality cohort genotypes that originate from GHK. So there is a bit of a caveat there, and we have to be careful in how we interpret our findings because of what we're defining as truth. And so to supplement this and enable us to make cross-species comparisons, we're using this additional metric, Mendelian inheritance errors or Mendelian discordancy. So when we look at across species, when we first start off with using the human model in the genome in a bottle sample. So up here, the two top plots have human samples with human models, and the bottom 2 plots are cattle samples with the default or existing versions of deep variant. On the x-axis, you can see F1 score, which varies or can change between magnitude of which changes, but on the y-axis, you will see the exact same deep variant models, starting with deep trio here, which is a joint caller, and then the deep variant allele frequency model, which just supplements this default version of deep variant with an additional channel. So what we can see though is that with cattle, we are not getting as great of a F1 score. So this is not what we had hoped by just being able to use deep variant straight out of the box. So we started to ask ourselves, can we do better by retraining with cattle? But before we could even consider doing this, the first step was drastically improving our ability to call variants and identify truth variants. So to do this, we had to go back to VQSR and optimize it for our cattle data set. So when we did that, what we did is we're able to reduce the number of Mendelian inheritance errors. So the orange bar, you can see our optimal version, and the gray bar is the sort of cattle standard based on the 1,000 bulls run 9. And so additionally, our strategy with how we created TrioTrain was that if we train on these trios, then we can remove those errors entirely and thus boost the accuracy of our truth labels. And here is just a very quick look at how this iterative training process works. So everything starts with a model checkpoint. So for the manuscript that we have in progress, this is the default version of deep variant. So then we give it examples from one of the parents in the trio, and then we evaluate how well that model can correctly genotype variants in the offspring, and select a new model based on F1 score, which then becomes the starting point for the next iteration, which then uses examples from the second parent in the trio and evaluates in the same offspring. And this is repeated across all parents in as many trios as you'd like. And each of these selected new checkpoints is tested by calling variants with this custom model and then comparing that against our original labels from GHK. And the way that we built this automated process was designed to be species agnostic. So as long as the input data are there and it's in the right format, then everything should work. There's no like, dash cow version of the TrioTrain pipeline. So we also were very strategic in how we organize the training of the model. So right here, I've just included what the initial models or existing models used, but then we had lots and lots of cattle data, so we were able to break it up over multiple phases. So the first two phases are kind of a cattle-specific version of deep variant, and then phases 3 and 4 were where we started to approach this idea of creating a multi-species or a golden model. So for phase 3, we actually include some bison trios. And then phase 4 is really key because we're using these F1 hybrids or interspecies crosses between cattle and outgroups. So what I do want to highlight here is that one of the key changes that happens across these phases is the heterozygous to homozygous alternate ratio. So here you can see F1 score again, and how it changes across all of the different iterations, specifically in the first two phases. So each line on the x-axis is just a different parent iteration over the training period. And then F1 score is on the y-axis. So what we begin to notice is that when we have iterations where we are training on non-reference cattle breeds, we actually have a bump in performance. And we are proposing that this is due to having more homozygous alternate variants and that the model is able to better identify those. Then we do notice that these indel performances in non-reference or in reference specific trio iterations kind of has a deviation, but it's kind of expected given the fact that we're relying on GATK, and thus our truth labels are not as perfect in indels. And then during phases 3 and 4, we kind of have this counterintuitive idea. So in deep learning, everything is always more data is always better. But actually if you're trying to game the system or trying to target a certain variant class in order to improve performance for that variant class, we actually see that that doesn't help. So when we start training on bison, that increases the number of homozygous alternates relative to the reference, but we actually see a decrease in homozygous alternate performance, which is then recovered when we move to these F1 hybrids, which have a high number of heterozygous genotypes and fewer homozygous alternates, and then we are able to recover that homozygous alternate performance. But for comparison and just sort of summarizing, we used the terminal checkpoints from each of these 4 phases. And here you can see the performance that we observed with the same testing data sets or the same cattle genomes, but we're comparing the 3 existing deep variant models again against each of these 4 checkpoints. And what you'll see is this drastic improvement in SNPs. And we do have quite a bit of improvement with indels, although not to the same extent as SNPs, again, due to imperfection in our truth labels. And then to sort of make true comparisons or apples to apples between different models and different species, we start using Mendelian inheritance errors. So on the x-axis, we have 3 different F1 hybrid trios. And then on the y-axis, you can see the rate of Mendelian inheritance errors for SNPs. And so the lighter green shade here that I'm highlighting is for the deep variant default model. So if we had not tried to use any of the custom versions and just used the recommended version, we would have an order of magnitude more errors, Mendelian inheritance errors in cattle than if we had performed retraining. So this equates to about a 36,000 reduction in errors per genome. And just as a final, to convince you that deep variant is really superior, if we had just stuck with GATK prior to doing any additional filtering that we did to undergo or create our truth labels, we would have had a much, much higher rate of Mendelian inheritance error. So even the default version is still quite a bit better than GATK, but obviously pursuing retraining improved that further. And what became very exciting for us is that we took the exact same models, but looked at, again at the Mendelian inheritance error rate, but this time in the genome in a bottle trios. And what you can see is that our cattle model, so after training in cattle, we call variants in the human genome, and we have half the number of Mendelian inheritance errors. So by retraining and giving new context, we're actually able to improve the model further, even in humans. And we're able to do that without joint calling. So down here, you can see the value or the number of errors with deep trio, which again is a joint caller taking 2 to 3 times the amount of time in order to generate the same results. And we're actually beating it slightly. And this was very exciting to us because we're like, finally, where you can have first evidence of our ability to make this species agnostic generalizable model. And I just want to emphasize that deep variant is not aware that I have given it cows, and it's not aware that it was given humans to begin with. So what it is detecting or picking up on are these variant class differences. So because we are changing the class distributions relative to the prior, we're able to give more context to the model and able to improve performance. And this is important because we already know of colleagues who have already started adopting the default version of deep variant. Typically, most of the published work has been focused on what we would call small sample size, so anything under 100 samples. And so for these types of groups, maybe pursuing retraining is a lot of effort for, maybe not as much of an impact. But for us at the UMAG group, we are always working with population sizes or population scale data. So for our 9,000 cattle genomes that we have, this would equate to an error reduction of about 330 million variants. So lastly, we were also very forward thinking in why we need a ability to retrain models. So we're part of the Bovine Pangenome Consortium and the Ruminant Telomere to Telomere Consortium, both of which are increasing the number of long read sequence data sets rapidly. And so most of those technologies rely on either deep variant or some other deep learning technology. So we need tools like TrioTrain to give us creative ways to customize these technologies for multiple species. For me personally, I want to take this model that I made with cattle and further validate it by using some of the data that is being generated as part of this Ruminant Telomere to Telomere Consortium, and sort of maybe gauge how well this model might work in these smaller niche species that don't get the same amount of attention that cattle do. And then, as Josue mentioned, my postdoc is primarily focused on developing the first genome in a bottle resource for non-human species. So we've already collected a couple cattle trios and generated primary cell lines. And then this fall, we're going to start sequencing. And then once we have that data, we'll be able to generate or improve how we curated the truth labels by including long read indels. And then when we have these higher quality labels, we'll be able to then go back and reuse TrioTrain and just improve the model even further. And again, we are not just interested in cattle, so cattle are just a starting point, and we're going to use this as a roadmap for other species. So we already have other collaborators that have reached out, such as your group, and primarily our expertise is focused on the animal side. So either our data for the UMAG group, and then we also have collaborators that have reached out to us about sheep, horses, chickens, and cats. I did want to highlight that we do have quite a few honeybee genomes. That number has taken off in the last couple months. So in summary, we find that exclusively training with human genomes restricts the application of these deep learning approaches for comparative genomics. And also, we were able to learn a lot by retraining with cattle. And the question becomes, how much better could we do with these genome in a bottle, high quality truth labels for all these different species? And fortunately, with how I built TrioTrain, our only limit is the quality of our truth labels. So I have many people to thank, including my lab and our collaborators at University of Idaho and Genome in a Bottle. And I want to just pause here. So this concludes the AGPT ag portion of my talk. And then next we can discuss more of the methods if there are any questions. Move you guys. All right. So a lot of the documentation or how my TrioTrain component is organized, or I have the GitHub page, which is available in the preprint that I believe Hostway has. And then I've created this wiki page for walking people through how to retrain or how to run TrioTrain using the complete human genome. So you go through here, it would walk you through a full start to finish genome in a bottle tutorial. I'm not going to do all of that for today. I just wanted to highlight it, that I do have resources for you to come back to this. But before that, I wanted to sort of a push for why you might want to switch to deep variant. One of the things that is really nice about using it is that it generates this interactive summary file for your call sets. So everything is interactive and scrollable, and you can sort of get at some of these quality metrics and actually look into all of this without having to parse a potentially large VCF file, which is pretty nice. But for starting TrioTrain, the way that I have it organized is that I just use a generic Excel file. So this would probably look different for something with the cranberries and just having a couple genomes. So each row in this Excel file is a trio. So then it's organized by like pedigree. So I include the reference here. And then if you have a population VCF to include, that is what triggers the inclusion of the allele frequency channel in the model. And then the pedigree is primarily right here. These run names, so that's just what I'm labeling and calling the trio name. And then this is where all of the actual initial data that is required. So for each of the trios, you need or each of the individuals in the trio, you need a BAM, BCF, and a callable bed file, which we primarily get through our GATK process. And then the minimum number of test genomes that is required is just one. So 4 genomes would get you the ability to run TrioTrain. Obviously, you can expand that to further. So just by increasing the number of columns, you can add more test genomes, and you can expand into multiple iterations or multiple trios by adding additional rows. So with that, I will do more of the computational tutorial. So I do everything on the command line. I'm not sure what everyone is familiar with, but for us, we're all on our HPC cluster. So let me go ahead and log in here. And then I created a little screen session for us. So this right here, the directory that I'm in is just the git clone directory from GitHub. And then I have template scripts for how I operate and what the pipeline expects. So to make things to basically smooth the transition. So if you're working on a Slurm-based cluster or an HPC, all of this should be familiar, or just be a polished version. So I start an interactive session, which just basically gets us off the login node and able to actually run things. And then some of the tools or the dependencies we already have pre-installed as HPC modules, and some other background helper scripts that get loaded with this script. All of that, again, is explained through more in depth in the walkthrough, like the actual tutorial. And then gotta always start the conda environment. So one of the key variables that are in the modules is this is how we determine which variant or which version of deep variant to use. So currently they're up at 1. version 1.6 of deep variant. I'm currently still using version 1.4 because version 1.5 is approximately the same for the short read sequencing model. There are some major functionality differences with the latest version, and there's been quite a few bugs over the past couple months. So until all the issues quiet down on GitHub, I'm sticking with 1.4. Then it's as simple as just running a Python command. So I also have a bash helper scripts to walk you through how to download the deep variant container and how to actually build the Conda environment. It can be a little bit particular as far as building the dependencies because deep variant uses a tool called Apache Beam, which requires a very specific version of Python that interferes with other things. And then there are other companion tools like Happy, which is a tool for variant comparison that is still using Python 2. So a lot of interdependency issues, but there's a lot of functionality that I've created with TrioTrain in order to give enough flexibility and with the idea that you're working with lots and lots of trio iterations. So for like a 1 or 2 trio situation, you probably won't need all of this, but you can see more of it here within the help. So I did want to, the cluster that I'm working on right now is very heterogeneous, so we did not include anything like SnakeMake or Nextflow, mostly because of issues specifically with SnakeMake in our Slurm cluster. Ideally, I would optimize all of this with Nextflow too. It's just that introduces a lot of bugs, and just trying to get the dang model built takes a lot of effort. So it's not currently using Nextflow or anything else. And the way that I control all of this is through a resource config file. So what I do is break up. I mentioned that there are different components of deep variant that are not optimized for GPU. So those phases are like make examples where it's actually creating the labels, the beam shuffling, Apache Beam, this reshuffling component that we added in order to get Beam to work on our cluster, and then the train and evaluation component. So for currently with the version 1.4, the kind of limiting technical issue is that it requires 2 GPU cards on the same machine. So figuring out how to get that to work on your architecture is kind of, it takes quite a bit of effort. So I don't know how much experience you have with GPUs, but a lot of that will depend on how your system works. And then it will go through the rest of the phases. So all of these different components under the different phases, those are just SBatch values for Slurm, if you've used Slurm before. Then to sort of start the actual retraining process, it takes that CSV input or the Excel file as a CSV as the metadata. And so to run it, a lot of times I use the dry run flag because it will go through everything and make sure everything is working for you. It will also allow you to look at the SBatch jobs that are about to be created, and you can sort of decide if things are actually working the way that they should be without fully committing to running thousands of jobs and GPU and all this other stuff with a typo or something like that. So the way that I run it is just TrioTrain and then provide that metadata file of the trios. So right now I'm currently in the process of training a model without a population allele frequency model. or the allele frequency channel for our collaborators who want to do polishing for some of these assemblies that they're building. But then the next thing that you have to set is the first genome. So right now, everything is based on a pedigree. So, but it's not basically whatever file or sample you put in the father label, that is what first genome is looking for. It's not necessarily that it will only take samples that are the father of the evaluation genome. It's just a way to structure training between two genomes iteratively, if that makes sense. So we're going to start with a father, and then the second round we'll take the mother column of the metadata file, and then create a name for the experiment. And then this is that resource configuration file that I walked through before. Define the number of tests. So this is just a check that the way that you've created the initial Excel file actually matches the expectations, that you haven't like thrown in an extra column or anything weird. And then, so I've already been retraining some iterations. So this is just starting the next trio. So I've, for the no-pop BCF model, I've gone through trios 1 through 8. Now I'm going to try running trio 9. So to do that, I do a start iteration, 17, stop, 19. So each trio is 2 iterations. So it's to run through both parents and the offspring requires 2 iterations. And then I'm just, for the sake of things, going to do the dry run. So it won't actually submit any SBatch jobs for me, but it will walk through everything and tell me what all it's going to be doing. So it will, right now it's where I talked about the number of variants can drastically influence the time for retraining. So the way that we set that is by looking at the number of variants in the truth VCF. So if you have 100 variants, then it's not going to take as long as, say, 24 million. So that is sort of how we've dealt with the fact that we're going to have a lot of intersample variability as we training. So right now it's gone through creating the jobs for both the father and the child, and now it's on the mother, and it won't create duplicate jobs for the child. So it will use the examples that were generated previously. It just runs a second iteration or a second evaluation with those samples or those same variants. And it will basically go through all of the different phases. So it will generate labeled examples. It will then shuffle them further in order to remove linear genomic order and go through the training and evaluation process. And everything it's very simple to avoid workflow management issues. Everything is just using Slurm dependencies. So certain components are happen in parallel, and then when that group of jobs, say all of the examples for father get completed, and all of the examples for the child are completed, then the training job is able to start. So technically it's rather unsophisticated, but this is what works for us. And so it's worked so far. Hopefully, as more people use it, we'll come up with perhaps more sophisticated ways of managing all of the work. But for actually working with the results from running TrioTrain, By default, there is a parameter to change where it writes outputs, but everything is basically structured here in this directory. So it will just go up one and start storing everything outside of the GitHub directory. So here you can see all of my different experiment versions. And one of the sort of unique things that we did, so this right here is the no pop model that I was explaining or actually running. So I've run it through Trio 8 and was about to start Trio 9. But the unique kind of way that I keep track of all of these different parameters and how things are changing across this like multiple experiments is all through the environment files. So just here. Yep, there you go. So everything, any of the environment variables or any values that get calculated as part of the training process, everything gets written down in a similar structured format. So I never have to question, like, did I include a pop BCF in that run or not? Did I change the number of epochs or the batch size, whatever? All of that gets written down. And so when it's adding new parameters, a lot of it gets just appended toward the end. So like it'll calculate the number of truth variants and decide how many of the regions it's going to create for shuffling. All of that gets written down along with the actual number of the training examples that were created before training went. And then along with what checkpoint it started with and where it ended up and across both of the iterations. So again, father does not have to be the father of the evaluation genome, but that's what we refer to it as. Then the actual outputs that get generated are all structured under the run name column. So however you want to organize it, if it's plant one, plant, I don't know, like you could call it whatever you'd like, but it will just organize it for you. So the examples that are generated here, let's actually look at a completed one. So this is going to be a lot of data. I apologize for all of that. So it's going to create lots and lots of files. So each region is going to get all of these shards and files for examples, and those get shuffled and consolidated because you're working with lots and lots of variant calls. Then the other kind of data that, so the examples get created first and then they get shuffled, and then training is able to start. So we can go through train. So this is where all of the actual checkpoints and time points that get written during the training process get written. So once it's identified and selected a best checkpoint, that gets stored as a variable, and then that becomes the new input checkpoint for calling variants, which is all here under test. So again, all we're doing is the one-step variant caller with deep variant. We're just using whichever custom checkpoint got selected during training and calling variants on all of the test genomes. And then we use the tool happy to determine or compare whichever the call set against the labels that we originated with GATK. So, and then process lots of different metrics from all of that to try to actually calculate some of those summary metrics that I included before. So, last thing I was going to mention was that SBatch jobs get written here. There's going to be a lot of them. So TrioTrain is doing a lot of work and generating a lot of files for you, but I've structured everything in a way that will hopefully allow you to navigate and find all of that as you if you want to customize any of this. Then the way that you sort of summarize post-training, because again, this is a lot of data to go through at once, is through the summary directory. So here, so after each one of the training rounds, TrioTrain will process across all of the evaluations that were done and sort of summarize them. So you're able to sort of compare without having to sift through all of this at once. So this is where some of those, again, I apologize for the formatting, but like you can start to see the false negatives and the false positives and the total count of homozygous variants and all of that, but look at it across all of the checkpoints that were generated or evaluated, I should say, during the training process. Then another cool thing that deep variant allows you to do is look at time. Oh, sorry, I realized I wasn't looking at time. So I'll go ahead and stop here because there's more that would take a lot longer. So there is a way to interactively look at the training metrics through TensorBoard. So I don't know if anyone's familiar with doing all of that, but that's a lot of how I decide whether or not something has, if I've had a good training run or not, and whether or not I need to maybe go back to the drawing board on how I've generated the initial data and try to strategize better. So that is probably a good stopping point. So apologies for how quick everything went, but hopefully that will help you get more familiar with TrioTrain and you can look into trying to use it.